After the vascular events, the next important events that we are going to go to in inflammation, if you can recall, we had discussed, these happen to be the cellular events. And we had already discussed that which cells will we focus our discussion upon. Those will be the white blood cells or the leukocytes. So the next thing that we will begin with are the cellular events. Now, unlike vascular events, in this we won't make a list first because this is a slightly more extensive list. So we'll first try to formulate an imaginary flowchart in our brain. We'll make an entire story and a flowchart, and right at the end of the topic, we will then come up with an entire summary of what are the cellular events that take place. Before we move on to the cellular events, there is one thing or one normal aspect that I want you to understand. Now, for example, this was the blood vessel that we were dealing with till now. Now, there is one important thing that you need to know: how is the blood flowing inside a blood vessel? So, remember, all the cells, for example, these are all the RBCs in red, and also the WBCs like the neutrophil, the lymphocyte, and so on. You can say all the cells are going to flow in which compartment? In the center. In the center of the blood vessel. Whereas what flows at then the periphery? Remember, at the periphery or the margin of the blood vessel, we have the plasma. So the cells are going to be at the center of the blood vessel, and the plasma is at the margin or the periphery of the blood vessel. This particular arrangement is followed in our body. This is how the normal blood is flowing in the body, and this particular arrangement is known as the normal axial laminar flow. Normal axial laminar flow. Axial laminar flow. This is normal. Now what happens is, remember we discussed one hallmark, that is increased vascular permeability, in which all the plasma was coming out of the blood vessel. So students, if I again draw a similar diagram, that if this is the blood vessel, in which now the lining endothelial cells, all the plasma has flown out. The plasma that was at the periphery, that has gone out because of increased vascular permeability. What will then happen to the cells? The cells now tend to come to the margin. Normally the cells were in the center, plasma was at the periphery, but from the peripheries the plasma has gone out. So the cells in turn now come to the margin, and students, this particular step becomes our first cellular event, and it is known as margination. So it is from the name itself, cells coming to the margin or WBCs coming to the margin, that becomes margination. So the first cellular event is margination. Please recall, what was the first vascular event? It was vasoconstriction. Overall, which is the first event? It is also vasoconstriction. So now the cells have come to the margin, we call it margination. The next step is a very interesting one, that is known as rolling. Means that again, if we have the blood vessel and the cells that have come to the margin, to the end, these are the endothelial cells, and the cells have come to the margin to the endothelial cell. What is going to happen over here? This cell is going to roll. It will roll, come to the next endothelial cell. Over here it will roll again, then come to the next endothelial cell, so on and so forth. So this is rolling over the endothelial cells. The WBC or the leukocyte is rolling over the endothelial cell. So this is known as rolling. Now, although it is called as rolling, I want to add on, and we are going to modify this event a little for our own understanding and for our memorizing purposes. We are going to call it rolling and weak adhesion. Rolling and weak adhesion. What do I mean by that? Let's take a magnified image of this area. If this is the endothelial cell and this is the leukocyte or the WBC, which is rolling, right? Not only is it rolling, but whenever it comes in contact with the endothelial cell, for a very brief moment they are going to adhere. But because the pressure of the blood above it is so much, it is going to detach and then go on to the next endothelial cell. When it comes to the next endothelial cell, again for a brief moment both of them will adhere, but the pressure of the blood is so much, it will again roll on to the next endothelial cell. So I can easily call it rolling and adhesion, but weak adhesion, because it will quickly detach and go on to the next cell. The question, students, that is asked over here is that this rolling and weak adhesion is mediated by which molecules? And remember, those are known as selectins. So remember that rolling is because of selectins. Very important. Now selectins, something that you need to mug up, they are also known as CD62. Students, throughout pathology a number of CD molecules are going to come up. Some of them we've done up till now in the chapter of cell injury, like we had a very important CD marker for apoptosis, that was CD95. Today we had done one CD marker while we were discussing endothelial cells. That endothelial cells marker is CD34. One more CD marker that we have added is CD62. This is a CD marker for other name for selectins. Now what are selectins? Selectins are something that are helping us in this rolling and weak adhesion. Remember, students, selectins are of three types. You have to remember three alphabets for selectins. These include P-selectin, E-selectin and L-selectin. PEL. P, E and L. Three types of selectins that we have. Indirectly, if the question says CD62P, CD62E or CD62L, means the same. P, E and L and 62 for selectins. The question comes over here because this is a slightly confusing concept. First of all, if you focus on the term E, why is it called E-selectin? Because this selectin is present on the endothelial cell. So E for E-selectin, E for endothelial cell. The next is P. I can also write P as the way you say P, PE. So this is present on the platelets as well as the endothelial cell. So the way you pronounce P, that is PE, platelet and endothelial cell. The last one that we have is L. L-selectin is present on the L for leukocytes. So three selectins and their location is simple to remember from their name. E, endothelial cell. P, so the way you say P, PE, platelet and endothelial cell. L for leukocyte. One very important concept, also something that you need to mug up a little, but we'll try to simplify this. Let's take two cells in interest that we have. One was the leukocyte and the other was the endothelial cell. So this is the endothelial cell and this is the WBC. When they are rolling, when the cell is rolling over the endothelial cell and weak adhesion happens, it happens because of selectins. I just told you that on the endothelial cells we have two types of selectins. One are going to be the P-selectin and the E-selectin. They are present on the endothelial cell. Whereas the L-selectin is present on the leukocyte. This is what we've done up till now. Now when binding or adhesion between two things has to occur, and if one of them has a molecule, the other one must have a corresponding molecule, only then will they combine. So what do I mean by this is that if P and E selectin are present on the endothelial cells, what is their corresponding molecule that is present on the leukocyte? That is something known as a sugar, that is Cellular Lewis X. Cellular Lewis X. So please remember P and E, you will always remember by the term PE, endothelial cell. But what is their corresponding molecule on the WBC with which it will bind? Cellular Lewis X. Similarly, go on to the other aspect. What was the selectin present on WBC? L-selectin. So this means that there is something corresponding present on the endothelial cell also, only then will it combine. And this is known as glycan, also known as madcam, also known as CD34. Remember, CD34 was a marker of endothelial cells. So obviously it will be present on the endothelial cells. So this is that particular table of Robbins that students tend to fear the most, but you just need to remember this diagram. P and E from the alphabets, endothelial cell, their corresponding molecule on WBC is sialyl Lewis X, a sugar. On the other hand, L for leukocyte, so L-selectin is on the leukocyte. What is it binding with corresponding on the endothelial cell? It is the CD34, also known as glycan or madcam. Now this is one of the most important tables that you need to understand. Remember, recall, selectins were CD62. Now students, there are one or two more questions that you need to finish off over here before we proceed. We just spoke about the endothelial cells having two types of selectins, P and E, right? There is one recent favorite question that has come up, and that says that if this is the endothelial cell, within the endothelial cell there are certain bodies that are present, and these bodies are known as the Weibel-Palade bodies. These are known as the Weibel-Palade bodies. Students, very important, Weibel-Palade bodies. First of all, you need to know where are the Weibel-Palade bodies present. They are present in the endothelial cells. What is their composition? What are these bodies containing? So, if you just focus on the two letters, Weibel-Palade, W, P. So, P will stand for P-selectin. P will stand for P-selectin, and W will be the Willebrand factor. Everyone knows that in the clotting mechanism, von Willebrand factor has a very important role to play. So von Willebrand factor and P-selectins actually have their source where? In the endothelial cells. Where in the endothelial cells? That is the latest favorite question, that is in the Weibel-Palade bodies. Please remember this. This is one of the recent favorite with the examiners. So students, the first cellular event was margination. Cells have come onto the margin. The second cellular event is that they start rolling over here with very weak adhesions. The third cellular event, as they roll, they now go into the third cellular event, and that is known as adhesion. That is known as adhesion. Now remember that adhesion was happening alongside rolling also, but that was weak. So I am going to, for our understanding and for our simplicity, we are going to call it firm or strong adhesion. So what happens that while rolling onto a blood vessel, if this was the blood vessel and these were the cells that were rolling, while rolling, as they keep rolling, there comes a time where they are going to stop. They are going to stop. After this, no matter what pressure of blood moves over it, it will stay there. Why? And why has it stopped? It has stopped because of firm adhesion. Adhesion between which two cells? Between the endothelial cell and the leukocyte. There is adhesion. So, there is adhesion between these two that are present. Now, if you remember, rolling and weak adhesion was because of which molecules? You are right, that is selectins. This firm adhesion that we have is because of molecules, that is integrins. So, rolling, selectins, adhesion, integrins. Just a very simple mnemonic before we proceed. Everyone knows about senior resident, senior residency. So S for selectins and R for rolling. So with this mnemonic, you will not confuse selectins and integrins. Selectins will be for rolling, SR, senior resident. Automatically then you will remember that integrins are for adhesion. And integrins are classified into two types, that is beta 1 integrin and beta 2 integrin. But over here the story is not as complicated as it was in selectins. The good news is that both of these are present on the WBC. So there is no confusion as to which is present on which cell. Integrin by definition is present on a WBC. So for example, if this is the WBC, if this is a WBC and this is the endothelial cell, this is a WBC and this is an endothelial cell. So where will beta 1 and beta 2 integrin be present? It will be present here. Say this is beta 1 and this is beta 2. Beta 1 and beta 2 always on WBC. There is no exception. But as we said, if two cells are attaching to each other, both of them should have some molecule, only then attachment will occur. So if WBCs have beta 1 and beta 2, what does the endothelial cell have? The endothelial cell also has two corresponding molecules. Let's read these. First of all, beta 1 integrin is also given the name VLA. As I said, there are certain things we need to mug up. Beta 1 is also known as VLA. But in our process of mugging up, we are also going to find out tricks to memorize. Since beta 1 is known as VLA, what is the corresponding molecule on the endothelial cell? This binds to V for VLA, V for VCAM. So beta 1, united V stand, united means 1. So beta 1, also known as V stand, so VLA, and what will it bind to on the endothelial cell? It will bind to VCAM on the endothelial cell. So I hope everyone remembers this now. United V stand, united means 1. VLA, other name for beta 1, binds to VCAM 1. Now, what is the other molecule? We had beta 2. Now in beta 2, the names given are, this is also known as CD11 and CD18. CD11 and 18. Now remember, what will this bind to? This will bind to ICAM on the endothelial cell. This will bind to ICAM on the endothelial cell. Now let's try to revise this on our diagram that we had made. So we had beta 1 and beta 2 integrins on the WBC. Integrins by definition always on the WBC. Beta 1 will bind to what molecule over here? Beta 1 is going to bind to united V stand, it will bind to VCAM. Beta 2 will bind to which molecule over here? It will bind to ICAM. I hope with this you are clear with selectins and integrins. Before we proceed on to the next, there is one link to microbiology and infectious diseases that I want to build up over here. A question regarding ICAM is important. Everyone has heard of cerebral malaria, which is caused by Plasmodium falciparum. Now, what is it that causes adherence of Plasmodium falciparum? Why is it that it causes cerebral malaria? Why is it going and attaching to the cerebral vasculature or the blood vessels or the microvasculature? What is mediating that attachment? So remember, ICAM1. ICAM is that molecule, is that cell adhesion molecule that is going to adhere Plasmodium falciparum to the cerebral microvasculature, thereby resulting in microvasculature, thereby resulting in cerebral malaria. So this is a link that we have made, slightly different from inflammation, link that we made to infectious diseases, that ICAM will be the adhesion molecule for Plasmodium falciparum to cerebral microvasculature, resulting in cerebral malaria. So this was about the first 3 steps that we discussed. First, cells coming to the margin, margination. Second, cells rolling with weak adhesion, which is because of selectins. Third, cells finally coming to a stop because of firm adhesion, that is mediated by integrins. Now what happens once the cell is firmly adhered? We come to the fourth cellular event. What is that? That if this is the blood vessel that we have with the endothelial cells, and now comes a point when the cell has finally come to a stop. It is adhered firmly over here. Now what happens is, slowly this cell is going to now come out of the blood vessel. Why? Because why is all of this happening? All of this inflammatory process is happening because there is some infection, there is some bacteria, some virus, some organism in the body which we want to fight, which we want to eliminate. So how is that going to take place? The WBC is going to come out and go and attack that bacteria. To attack that bacteria, it has to come out. It has to migrate out. So we are reaching that term slowly. If it has to migrate out through the blood vessel, what will I call it? This will be known as transmigration. This will be known as transmigration. So trans means through. So through the blood vessel, it is going to, migration means movement. So through the blood vessel, now the WBC is going to come outside. Transmigration, one of the favorite steps with the examiners. They tend to ask two or three very important MCQs, patent MCQs that you need to know, repeat one-liners which you cannot afford to get wrong. What are those questions? The first question: which are the vessels which will maximally show this process of transmigration? So maximum involved vessels. That is the first question. Remember, usually when we had done vascular events, what was the maximally involved vessel for vascular permeability? It was venules. Maximum permeability was seen in venules. Similarly, maximum transmigration is also seen in venules. But whenever, remember, we have a rule, we have an exception. So everywhere in the body, maximum transmigration will be seen in venules, except in the pulmonary circulation. Except in the pulmonary circulation, where maximum transmigration is seen in the capillaries. So maximum transmigration is seen in the capillaries. So overall, always it is venules, but for pulmonary, for the lungs, for the circulation in the lungs, it is capillaries. What is the next question that is put up? Like we had selectins and integrins, what is mediating this entire process? Remember, the most important molecule that we have for transmigration is known as PCAM1, also known as CD31. Again, another marker in our CD list has increased, but remember, this happens to be one of the most important because this particular question has been asked n number of times in the exam. For some reason, the examiners love this question. What is the molecule that is mediating transmigration? That is PCAM1 or CD31. Now students, transmigration is a self-explanatory term. Examiners tend to, instead of giving you transmigration, tend to give you the other term for it. Remember, transmigration is also known as diapedesis. Also known as diapedesis, right? So instead of giving you a simple term like transmigration, the examiner will give you a slightly difficult term like diapedesis, but don't get confused, they essentially mean the same. Now students, two questions you've done: maximum venules in the lungs, capillaries, molecule CD31 also known as PCAM1. But you need to understand a concept that if a cell is coming from inside to outside, it has to break a lot of barriers in between, like the basement membrane over here, a lot of extracellular matrix over here. Only then it can come out. It has to make its path, it has to pave its path and then come out. So this also needs certain enzymes, and this is mediated by certain enzymes known as MMPs. Full form of MMPs is matrix metalloproteinases. Matrix metalloproteinases, MMPs, and a very important example of MMP is collagenase. A very important example of MMP is collagenase. So, to break down the basement membrane is type 4 collagen. Break down the basement membrane, to break down the extracellular matrix, there is a lot of collagen present there. To break the collagen, what enzyme would be needed? Collagenase. The cells are going to release collagenase. These cells will release collagenase, will break down all of this collagen around it, will make its path, and then finally come out. So these three things about transmigration and diapedesis are very important. Now students, we've done 4 cellular events. The cell has finally come out. So you can say that till now whatever we were discussing was happening inside the blood vessel, intravascular events. Now what will happen outside the blood vessel in continuity. Those will now be the extravascular events. So now coming on to the 5th cellular event that we have. Again a simple one. The blood vessel, which we are now going to leave out because the WBC has come out. This is where the WBC is present, outside the blood vessel. This WBC, why has it come out? What was the main aim? Because somewhere in the body there is some bacteria or there is some antigen. There is, say for example, let's take example of a bacteria. So somewhere in the body there is a bacteria the body wants to get rid of, and to get rid of it is sending its WBCs. So it has come out mainly because it has to move towards this bacteria. Now this is our next step. This process is known as chemotaxis. This process is known as chemotaxis, right? So remember, if I want to put it in theoretical or definition, because very important is the definition, it is a one-liner question. Chemotaxis is nothing but the unidirectional. It is the unidirectional. Please focus on every term over here. Every term is an MCQ. It is the unidirectional and targeted movement. Of what? Of the WBC towards the organism. Why have I focused on these two terms so much? It is unidirection. The WBC can only go towards the bacteria or towards the organism, nowhere else. Will it be bidirectional, multidirectional? No. That is how the MCQ comes. You need to remember that it will always be unidirectional. That is exactly what the examiner tends to focus on. Second, it is targeted. It knows that it has to go to its target. The WBC knows this. It has to go to that bacteria. It has to go. It won't go anywhere else in the body. So it will be a very focused student, you can say. It knows its goal. It knows its target. The WBC is going to move towards that target in a single direction. Will not have any distractions in between, will not move haywire, it will go straight to that organism. So chemotaxis is unidirectional targeted movement of the WBC to the organism. That is most important. Now why? This question should automatically come. How is this happening? The WBC is free to move everywhere. Why is it that it is going only to that bacteria? What is its motivation that is giving it so much of focus in life? So remember that this is because of certain chemotactic agents. This is because of certain chemotactic agents. Now there are certain things over here, or you can say the chemotactic agents over here, which will make sure that the WBC goes nowhere but to the organism, right? So these chemotactic agents are going to create a gradient. They are going to create a gradient. And how will they create a gradient? what you are asked in the exam, what are the examples of these chemotactic agents? So remember, the chemotactic agents are of two types. They can be exogenous and they can be endogenous. Exogenous and endogenous. Exo means something from outside. So exogenous chemotactic agents, easy list because we have only one example to remember, thankfully, that is bacterial products. Bacterial products. The bacteria that enters the body disintegrates into products. Those products in itself, they ensure that they pull the WBC towards themselves. So bacterial products. Remember, the question comes is, which is the only exogenous chemotactic agent? So you need to know the only exogenous chemotactic agent is the bacterial product. Rest everything else will be from within the body, that is will be endogenous. How will we remember these endogenous agents? So for this I've got a simple mnemonic for you, that is LIC. What is LIC? L stands for leukotriene B4. L stands for leukotriene B4. I stands for interleukin 8. And C stands for C5A. Right? So remember, LIC, leukotriene B4, interleukin 8 and C5A. These three are very important. These are the endogenous chemotactic agents. So what is happening? These chemotactic agents are like your mentors. They are going to guide you towards a target. So these chemotactic agents are like mentors for this WBC. They will make sure that this WBC remains focused. They will make sure that this WBC goes nowhere else, knows its target and goes to its target, that is the organism or the bacteria. One question that you need to know, the last one about this chemotaxis, is everyone knows of drugs, that is steroids. So one pharmacological correlate that you need to know, something that we are going to drift away a little from pathology and go into one more subject that everyone dreads a lot, that is pharmacology. Everyone has heard of steroids. Steroids, also what everyone knows is that steroids have anti-inflammatory actions. Anti-inflammatory actions, but where all does it inhibit inflammation? There are multiple areas. One of them is that it is going to inhibit chemotaxis. So if the WBC cannot reach the bacteria, inflammatory process will come to a halt, inflammatory process will stop, and that is what steroids are acting. One of the mechanisms of steroids is that it inhibits chemotaxis. Now students, going back to this diagram, the WBC by chemotaxis has moved to this organism, that is the bacteria or any organism that was causing infection in our body. What this WBC has to move through is a lot of extracellular matrix. There is a lot of material in between. It has to break through that and it has to come to this. We know the enzyme that will break all the collagen, collagenase or MMPs, we've discussed that. But there is one receptor also that plays a role over here. One receptor that you need to know. So remember that if this was the WBC, and this is all the extracellular matrix through which it is moving. This extracellular matrix has something known as heparin sulfate. And this heparin sulfate will bind to something on the WBC known as CD44. Right? So WBC has CD44, extracellular matrix has heparin sulfate, they will bind. As it keeps binding, the WBC keeps on releasing its enzyme, that is MMPs or collagenase, and keeps moving forward. Forward towards whom? To the bacteria. What do you call that? We call that chemotaxis. So students, we've done 5 very important cellular events in which the WBC has finally reached the organism. Now we come to the 6th event that you need to know. The 6th cellular event that we have is something known as opsonization. something known as opsonization. Now, where were we? That if this was a blood vessel and the WBC had come out of it by diapedesis, the WBC had gone to the bacteria and this was chemotaxis. I hope everyone is in sync up till now. Now what happens in the next step is that on this bacteria there is one coating that is going to develop. So this is the bacteria, and all throughout we are going to cover the bacteria with something. That process is known as opsonization. So opsonization is nothing but the coating of the bacteria or the organism. Why are we doing that? First of all, why, and then how. For everything you should know these two questions. Why are we doing this? Because for example, there are 10 bacteria, just an arbitrary number. Out of them, 5 have undergone opsonization and 5 have not. Which out of these will your body kill? Remember, body will kill both, the ones that have undergone opsonization and the ones that have not. But preferential killing or a faster or a quicker killing will be of those which have undergone opsonization. So first of all, why would be because coating of the organism results in preferential killing. Coating of the organism results in preferential killing. Remember, this process is not mandatory. This is a question that has been asked, friends. This is not mandatory. Even if opsonization does not occur, it's not that the organism will never be killed. It will be killed, but at a slower pace, at its own time. But we want quick elimination. We want our body to be free, so we have opsonization so that they are preferentially killed faster. First, preferential killing. Second, it is not mandatory. Once you know the why are we going in for opsonization, the next question that comes to us is how. How is this opsonization happening? So for that, students, remember that if this was the bacteria or the organism and this is the coating that we have, this coating is now known as the opsonin. Coating is done by opsonin, right? Opsonins are of multiple types. What are the examples? The first opsonin is the FC portion of IgG. So we've come up with the long term. Everyone knows IgG is nothing but immunoglobulin G. What is this FC portion? Every immunoglobulin has 2 heavy chains, right? Everyone knows it has 2 heavy chains and it has 2 light chains. Everyone is well aware of this particular structure, 2 heavy chains and 2 light chains. There are 2 ends. This is one particular end of the immunoglobulin and this is the other end of the immunoglobulin. This end is known as the FC portion and this end is known as the FAB. What is FAB? It is antigen binding. That is, it is at this portion that the antigen binds to the antibody. So now if you know this structure, that there are 2 heavy chains, there are 2 light chains, there is one FC portion and there is one FAB portion, I am saying opsonin is FC portion of IgG. Means that which portion of IgG will bind to the bacteria? It will be the FC portion. This is how the binding will occur. The FC portion will bind to the bacteria. Now this was the first example of opsonin. The second example of opsonin that we have is going to be complement proteins. Complement proteins. These include C3b, C4b, C5b. And remember, in the same order. So the most important one becomes C3b. The third one that we have are going to be certain plasma proteins are going to be certain plasma proteins. These include example fibrinogen and CRP. Again, a very important molecule we've just put up, CRP. What is CRP? It stands for C-reactive peptide. It stands for C-reactive peptide. Now students, although we've elaborated this C-reactive peptide, there is one full form which no one usually focuses on. What is this C? This C is not only what we need to know for our concept building, but also what has been asked as an MCQ. This C is nothing but the capsular polysaccharide. It is the capsular polysaccharide, or you can say a carbohydrate, of which organism? So P for peptide, of pneumococcus. So P for peptide, P for pneumococcus. It is a capsular polysaccharide of pneumococcus. So that becomes the entire full form, C-reactive peptide, where C is for the capsular polysaccharide of pneumococcus. Now what you need to, one more thing before we proceed, something which links us to medicine. Have you heard of a similar term? This is C-reactive peptide. If I remove the reactive over here, what am I left with? C-peptide. So you must have come across a term that is C-peptide, and where have you come across this term? Yes, you must be able to recall that C-peptide is produced alongside insulin. So if 20 molecules of insulin are being produced in our body, 20 molecules of C-peptide are produced by our body. So C-peptide is totally different. C-reactive peptide is an opsonin, is involved in inflammation, that is totally different. We do not have to confuse these two. Let's take up one question of medicine. If we want to discriminate or differentiate that whether a person's rise in insulin is because the body is secreting more insulin or because the person has taken some exogenous source of insulin. In either cases, when you do an insulin testing in the blood, in either cases it will be elevated. How do I differentiate? To differentiate, I instead do the C-peptide. C-peptide will increase only if the body has produced insulin. If the body has produced 20 molecules of insulin, means the body has also produced 20 molecules of C-peptide. If a person has done some drug abuse and if the person has taken a more amount of exogenous insulin, then only insulin has been taken. There is no increase in C-peptide. So one very important question on medicine also we've just discussed. Coming back to our topic, remember we are not dealing with C-peptide, we are dealing with C-reactive peptide. So if you have to sum up, there are these three opsonins that we did. One, the FC portion of IgG. Second, complement proteins like C3b, all the Bs, 3b, 4b, 5b. And third, plasma proteins like fibrinogen and CRP. Now students, there are two important MCQs that are asked, have been asked and potential as well. Question number 1, which is the most important opsonin? Out of this entire list, which is the most important opsonin? So I made this list in the same order. The most important opsonin, remember your answer will be IgG. But please read the question carefully over here. Instead of giving you this question, they just added one more term in this question and they said, which is the most important complement opsonin? So question sounds similar. In both cases they are asking you important opsonin, but in first they were asking you overall, and in the second question they are asking you particularly complement. Complements were 3b, 4b, 5b, and I told you we had written them in the same order. So the most important complement opsonin will be C3b. So these are two very important questions on opsonins that we have done. Now once you are going back, the bacteria is now taken up or is coated with an opsonin. The WBC is also over there. All the raw material is ready for the final step of cellular changes or of inflammation, and that is the 7th step that we have for what everything was being actually carried out, and this is phagocytosis. Now, this is the final step for which everything was being done up till now, phagocytosis. What do you mean by phago? Phago means eating up, engulfing. Phago means eating up or engulfing. Cyto means cell. So, a cell is going to eat up the bacteria. That is exactly what we have. We have a cell, a WBC. We have a bacteria which is coated all around by an opsonin, and that cell is going to come and going to eat the bacteria. That is phagocytosis. I want you to recall one question over here which we had done right in the beginning of this chapter. Who was the scientist who gave us phagocytosis? And the scientist was, and I told you, as long the name phagocytosis, as long is the name of the scientist, that is Mechnikov. Now let's go into the mechanism of phagocytosis. So what is the mechanism that phagocytosis occurs through? We are going to divide it into three categories. If there is a cell and there is a bacteria, the first thing is that the cell must recognize the bacteria, that yes, this is the bacteria I want to eat. So the first step becomes recognition. The cell has recognized that yes, this is my target. Next, the cell has to eat up that target. So I will call it engulfment. And third, once the cell has eaten up this bacteria, our ultimate aim, why have we been studying so much of inflammation? Because we want to kill the bacteria, and the ultimate aim is accomplished, that is known as killing. So students first recognize, then engulf, and then finally kill the bacteria. This killing, students, can be of two types. Either it will utilize oxygen, so it will be oxygen dependent killing, or it will not utilize oxygen, so it is oxygen independent killing. Before we go on to the details of each one of these, what I want you to definitely focus on right now, if someone asks you, out of the killing mechanisms, which of these is more potent? So remember, it is the oxygen dependent one which is more potent. The oxygen dependent one is the more potent mechanism that we have. So this is a question that has been asked in the previous years. Now this is a basic summary of phagocytosis that we have. We are going to take each step in detail and understand this mechanism. So the first step that we had, that I told you in phagocytosis, was recognition. So for example, this is the bacteria and this is the WBC which is going to eat that bacteria. Say, take an example of a neutrophil. This has to recognize, these two have to attach, they have to recognize each other. That will be by certain receptors. This recognition is because of certain receptors. And what are those receptors? One is known as the mannose receptor. Remember, the bacterial cell wall has mannose residues. So it will bind to which receptor of the WBC? The mannose receptor, because there are mannose residues present in the bacterial cell wall. In cell wall of bacteria. First, mannose receptor. Next, there are other receptors also known as the scavenger receptors. Scavenger receptor you will definitely read in medicine and also in cardiovascular pathology. Scavenge means it is going to take up something, eliminate it and save us. So, the oxidized LDL in the body, which we do not want that should deposit, that oxidized LDL is scavenged by these scavenger receptors. If they deposit, what will it result in? Correct, coronary artery disease. So we do not want that, they are going to scavenge it. So we have scavenger receptors. Now there is a bacteria which will be recognized by the WBC because of mannose receptors, scavenger receptors. What was the next step after that? After recognition, the next step that it has to go into is engulfment. Now, how will that happen? So, if this was a particular neutrophil or the cell, and this is the bacteria which is covered by a layer of what? Correct, it is opsonin. They have attached to each other. What this particular WBC will do, it will send out certain pseudopods. Why is it sending out these pseudopods? Because it wants to engulf that bacteria and take it inside. So it is going to send out these pseudopods. One very important thing you need to know, this pseudopod formation is because of actin polymerization. So there is actin polymerization, pseudopods are formed, they are going to take the bacteria inside the cell. This bacteria now enters the cell. What happens when it enters the cell? So if this is the particular cell that we have, and now it has engulfed that bacteria, so that bacteria has come inside, and inside it is present the cell. This is known as a phagosome. When that bacteria has been brought inside the cell, that is a phagosome, right? Now all of us know that inside the cell we have an organelle known as the lysosome. So the phagosome has the bacteria, our cell has a lysosome. These are now going to fuse. The phagosome will move towards the lysosome. And ultimately these two are going to fuse with each other. So if this was the cell that we had, the phagosome, if this is the phagosome containing the bacteria, and this is the lysosome, both of them will fuse, and by fusing the two you also have to fuse the two terms. We will now call it a phagolysosome. So to combine the two terms, phagosome, lysosome, phagolysosome. Now remember, I told you that the phagosome has to move towards the lysosome. This movement is mediated by the LYST protein, the list protein, a very important concept because there is a disease associated with this. It is the list protein. Now finally the phagosome and the lysosome have fused, phagolysosome is formed. So moving on, why are we doing all this? Because we wanted to enter into our third step, that was killing. And if you can recall, killing was of two types, that is, either it was utilizing oxygen or it was oxygen independent. Students, please recall which out of these was the more potent one? It was oxygen dependent killing. So since that is more potent, we will discuss that in more detail. Now what we are going to do, first we will start with oxygen dependent killing. But at the same time, remember, we are taking a magnified view. There was a phagosome over here, it fused with a lysosome over here. Let's consider that the lysosome has multiple enzymes. And I'm just going to take this particular section, and I'm going to magnify this section. This part of this area of the phagolysosome I'm going to magnify for oxygen dependent killing. So for example, if this is the entire membrane that I wanted to, of that lysosome, this is the membrane. The first important thing, the membrane has an enzyme over here that is known as NADPH oxidase. NADPH oxidase. Now what do you need to know at the outset is, NADPH oxidase is the most important enzyme for phagocytosis. Question number 1. This is the most important enzyme for phagocytosis. Now NADPH oxidase, parts of this enzyme are present in the cytoplasm. Parts of this enzyme are present in the membrane. When the process of inflammation starts, both of these are going to fuse. The cytoplasmic parts also come to the membrane, and finally on the membrane we have a complete enzyme complex of NADPH oxidase. Again, a very important step because there is a disease associated with it. So remember, NADPH oxidase, which is the most important enzyme in our entire pathway, has some amount in the cytoplasm, some parts in the membrane, but when we started this process, all of it fused and came to the membrane. Now, it is utilizing oxygen. It is an oxygen dependent killing. So what does this NADPH do? It is going to first convert oxygen into a free radical, that is superoxide. After superoxide is formed, this is then going to combine with two molecules of hydrogen to form something known as H2O2. What is H2O2? It is hydrogen peroxide. If you can recall from cell injury, this process had a certain enzyme, that is SOD, superoxide dismutase. Very important enzyme that we had discussed when we were discussing reperfusion injury. Superoxide dismutase. H2O2 is formed. H2O2 is also a free radical. Superoxide is also a free radical. They can kill the bacteria, but still we are proceeding with the step further. Why? We want something which is so potent that kills the bacteria immediately in one go. So what does the body do? From this H2O2, in the presence of an enzyme called MPO, that is myeloperoxidase, as well as chloride. So H2O2, MPO, chloride. These three will combine and form something known as hypochlorous acid. Hypochlorous acid. HOCl, hypochlorous acid. Now students, what you need to recall from the days when you studied chemistry, HOCl was the chemical formula of what? It was actually bleaching powder. So in order to kill a bacteria, what are we forming inside the cell? We are forming bleaching powder, which is HOCl. Remember, this happens to be the most potent. That is why we are forming it. It is the most potent molecule that we have, because if the bacteria comes in contact with this, immediately this will result in killing. This will cause killing of the bacteria. Our aim is accomplished. So to revise students, this process started because of an enzyme, NADPH oxidase, which had a cytoplasmic part and a membranous part, and they fused. Most important enzyme, formed superoxide, which formed H2O2. H2O2 combined with two very important things, MPO and chloride. MPO stands for myeloperoxidase, and formed bleaching powder, which causes killing. Because of this entire process that we just studied, this pathway is also known as the H2O2 MPO halide system. Why? Because it uses H2O2, it uses MPO, it uses chloride, which is a halide family. So H2O2 MPO halide system. Indirectly, if they say, which is the most potent method of killing, oxygen dependent or independent? You know oxygen dependent. Oxygen dependent, if they don't write in the question or in the options and they write H2O2 MPO halide system, then you know that is oxygen dependent pathway. So you will opt for H2O2 MPO halide. I told you NADPH oxidase was the most important enzyme. If I am saying it is the most important, means that there are some other enzymes also definitely present. That is why it is the most important. So on the same membrane, on this particular same membrane, I am going to also consider one more enzyme, that is nitric oxide synthase. nitric oxide synthase. You know the term synthase. What is it synthesizing? Nitric oxide. That is why. But going back to our biochemistry review, linking it with biochemistry, nitric oxide is formed from which amino acid? It comes from arginine. Correct. It is arginine. So arginine forms nitric oxide. Nitric oxide then combines with this superoxide that we have to form something known as peroxynitrite. Peroxynitrite. And this peroxynitrite then causes killing of the bacteria. So we have two pathways. One, the NADPH oxidase pathway, H2O2 MPO halide system as you may call it. The other is the nitric oxide synthase pathway, which uses nitric oxide, converts it into peroxynitrite, which causes killing. Which out of these is the most important pathway, or which out of these is the most potent pathway? That will be NADPH oxidase pathway. Okay, now that we have studied this entire process, this is the, please recall, oxygen dependent killing. I want you to get back and understand that this was the oxygen dependent. We are left with one more form, that is oxygen independent killing. So what do you have to understand about oxygen independent killing? Remember, in oxygen independent killing, this is not as elaborate. This is because of certain molecules like lysozyme, the lysosomal enzymes, lysozyme, lactoferrin, something known as defensins. So how I've remembered it is defense mechanism, our body is going to defend us, so defensins will help in killing. Fourth is something known as MBP. Very important. In this list, an important question that has come up, MBP, students, is major basic protein. Major basic protein. Major basic protein has two questions that have been asked. What are those two questions? From which cell is major basic protein released? And I'm going to give you an indirect hint. What was the cell that had a spectacle shaped nucleus and had orange pink granules? Yes, that cell was an eosinophil. That cell was an eosinophil. So eosinophil is that cell which has these orange pink granules, and what are those granules going to release? This particular molecule, that is major basic protein. Now indirectly the examiner can twist the question and ask you that if eosinophil releases the major basic protein, what type of infections will major basic protein help you fight against? And if you can recall, we just studied that eosinophils are going to help us fight parasitic infections. Parasitic infections. So the eosinophil releases this MBP, which is an oxygen independent pathway, and that helps us fight against parasitic infections. The last in this list that we need to know is a newer term that has come up, something known as cathelicidins. Now this is one of the newer terms and becomes a potential question for us. Cathelicidins have two important concepts that we need to know. One, you already know that this is helping us in killing. But the first thing that you must memorize now is that this is vitamin D dependent. So if, let's take a situation that a person has deficiency of vitamin D in the body, such a person, will that person be able to produce cathelicidins? No. So cathelicidin levels will be low. What are the implications of this? This helps us in fighting against tuberculosis or the tubercle bacillus. So now fit this into our hypothetical situation. There is a person with vitamin D deficiency, because of which the levels of cathelicidins have gone down. If cathelicidins are going down, the chances of which infection will go up? That will be tuberculosis. So these two students tend to be potential questions that we are expecting in the coming exams. All of these, if we want to summarize, all of these were oxygen independent pathways: lysozyme, lactoferrin, defensins, MBP from the eosinophils, and cathelicidins, the recent and the most important one. So these form a summary of how the bacteria that had entered the body fought, and we fought against it by some vascular events, followed by some cellular events, and finally the WBC has come to the bacteria, engulfed it, and resulted in killing.